Welcome to a presentation of protein products of the company BioVendor. In the beginning, let me make a general introduction to our company. We are active on the field of biomarker research. We produce classical ELISA kits, but in addition to that, we also develop new innovative technologies. The most promising novel technology is Mireia. It's an immunoassay-based method of microRNA quantification. MicroRNA is considered to be a new important biomarker of many serious diseases. Please take a look at the video introducing our innovative Mireia method. Our company also produces components that are essential for immunoassay design, particularly proteins and antibodies. We have a long experience in protein production because we manufacture them for 20 years. During this long time, we developed hundreds of proteins. We use them for our applications, so our proteins must be of high quality. BioVendor manufactures both recombinant proteins and native proteins. Recombinant proteins are expressed in two expression systems, mammalian cells and bacterium E. coli. Both expression systems have their advantages. Mammalian cells produce proteins with excellent post-translational modifications, for example, glycosylation and disulfide bonds. Therefore, proteins expressed in mammalian cells are of high quality. The advantages of the E. coli system are lower cost, shorter production time, and easy upscaling of production. As for our native proteins, we isolate them from various tissues, like plasma, serum, urine, breast milk, and many others. In our company, the production of proteins is a starting point for the development of other products. We use our proteins mainly for the antibody production, immunoassay development, and immunoassay production. Many other companies and academic institutes find our proteins suitable for their research, so we sell our proteins to many customers. This is an overview of the development of a recombinant protein. It's quite a complex process, including many steps demanding on high professional skills of our staff. The vial on the left is the result of our effort. Our customers receive our proteins in such vials. Our products have many advantages. We try to avoid usage of additives and preservatives. For example, we don't add albumin to our proteins because albumin makes it difficult to determine the concentration and purity of the protein of interest. Most of our proteins are lyophilized. Lyophilized proteins are stable and can be delivered at ambient temperature. The usual quantity per vial is 0.0 milligram, but we are flexible to deliver other quantities according to the needs of our customers. We strictly control the quality of our products. Our quality control includes several methods, which will be explained later. We provide our customers with an extensive information about our proteins. Customers receive large product data sheets, and all the information can also be found on our website biovendor.com. Another great advantage is a careful technical support for our customers. All questions and inquiries are answered by our specialists. Last but not least, we provide our customers with interesting discount offers on all our products. As I mentioned before, our quality control is very tight. This is an example of a certificate analysis where the results of all quality tests are stated. Our quality control includes several following steps. We determine the protein concentration by the BCA method. We carry out the SDS page electrophoresis to visualize the protein and evaluate the purity of the protein. We perform gel filtration chromatography to assess multimeric structures. We measure endotoxin levels. We use mass spectroscopy to confirm amino acid sequence of our proteins. and we carry out stability tests after dissolving the lyophilized proteins. We provide our customers with detailed information about our proteins. This is an example of a product data sheet. It has several pages and contains extensive information about the protein. Our customers receive all information important for their applications. Product data sheets and certificates of analysis are delivered with our proteins. They can also be downloaded from our website. Now let me introduce some of our most interesting proteins. For example, Adiponectin is a promising clinical biomarker of many diseases like metabolic syndrome or atherosclerosis. A specific quality of this protein is formation of multimeric structures. We offer several types of adiponectins. For example, adiponectin rich in high molecular weight multimers or adiponectin forming a trimer. We also have a fragment of adiponectin containing only the globular domain. We also offer native adiponectin and various animal adiponectins. We established a test for the biological activity, and we can state that our adiponectins are biologically active. A lot of our customers are scientists using our proteins in their research, and they refer to our proteins in their publications. I chose an example of a reference in a journal with very high impact. This research laboratory used our adiponectin to investigate its vasodilating effect on endothelial cells of arteries. Many other references to our adiponectins can be found on our website or in the product datasheets. Our human lipoprotein lipase is a recombinant protein expressed in mammalian cells. Lipoprotein lipase affects the lipid metabolism, so most of the customers are researchers studying diseases related to the lipid metabolism. Again, we can cite scientific articles referring to our lipoprotein lipase. In the displayed example, our lipoprotein lipase was used to assess the cross-reactivity of antibodies. Our human clusterin is a native protein isolated from human plasma. Clusterin is connected with neurodegenerative diseases and many oncological diseases. Clusterin also works as a chaperone. It stabilizes protein folding. Our clusterin is referred in scientific publications, and I chose an article on research where clusterin was used to explore its effect on the conformation of cathepsin K. Our human omentin is expressed in E. coli. Omentin is studied as a possible biomarker for acute ischemic stroke or colorectal cancer. Our omentin was used in a lot of scientific works. I picked an article of a research group that used our omentin in animal studies to explain its biological functions. Our Stanniocalcin-1 is a recombinant protein expressed in mammalian cells. It stimulates tumor growth and is studied as a possible oncological biomarker. Our Stanniocalcin-1 is also referred in many scientific publications. I selected an article of a research group that used this protein in cell culture experiments exploring its effect on apoptosis. Our uromodulin is a native protein isolated from human urine. Recent scientific studies reveal that the biological function of uromodulin includes protection against urinary tract infections and protection against kidney stone formation. Uromodulin is also involved in blood pressure control and innate immunity processes. Again, I chose an example of a reference to our uromodulin. This research laboratory used this protein as a standard for quantification of uromodulin. A lot of our customers request our company to produce specific proteins of their interest. So we offer a service of custom protein production. Our advantages our wide experience in protein production, low price, short delivery time, and high quality of our proteins. Our customers can consult their specific demands with our specialists. We are able to produce various kinds of proteins according to the needs of our customers. Thank you for your attention. I hope you find our presentation useful. If you have any questions, feel free to contact us. And please visit our new website, biovendor.com. Thank you.